Cells communicate using various signaling pathways, and one key pathway is the IP3 signaling cascade. A signaling molecule, such as a hormone or neurotransmitter, binds to a GPCR on the cell membrane. This binding causes the GPCR to change shape, activating it. This activated GPCR then interacts with a G protein. It causes the alpha subunit to exchange GDP for GTP, which activates the G protein. The beta-gamma complex of activated G protein dissociate from alpha subunit and move along the membrane to activate an enzyme called phospholipase C-beta. The activated phospholipase C-beta interacts with phosphatidylinositol 4,5-bisphosphate in the cell membrane. Phospholipase C-beta then cleaves IP2 into two key molecules, diacylglycerol and inositol triphosphate. Diacylglycerol stays in the membrane and acts as a docking site and recruits protein kinase C to the membrane, while IP3 moves toward endoplasmic reticulum. When IP3 reaches the ER, it binds to the IP3-gated calcium release channel located on the ER membrane. This binding opens calcium channels, allowing calcium ions to flow from the endoplasmic reticulum into the cytoplasm. As calcium ions are released into the cytoplasm, their concentration increases rapidly. The binding of the calcium with protein kinase C activates the enzyme. This enzyme is now ready to phosphorylate the target proteins. These phosphorylated proteins initiate various cellular responses, depending on the cell type, such as gene expression, metabolism, cell differentiation, or growth. When the calcium level rises, the calcium ions bind with the calcium-binding protein called calmodulin and form calcium-calmodulin complex. This complex interacts with various target proteins to regulate cellular functions. Calcium also acts as a secondary messenger. High level of calcium in the cytoplasm play a critical role in activation of enzymes, muscle contraction, gene expression, and metabolic regulation.
Please like, share and subscribe, and don't forget to hit the bell icon for more videos.